So, back in the day, it was presumed that fungi were some sort of degenerate plant form. After all, mushrooms not only look vaguely plant-like, they, together with all the other fungi like molds and yeasts, they have cell walls, and this makes them more plant-like than animal-like. But then came molecular sequencing, and even from the earliest and most unsophisticated days of molecular analyses, it was starting to look like the fungi were actually closer to animals than they were to plants. And this seemed totally weird. Maybe it was some kind of mistake or faulty interpretation of the data. It's not easy to convince the scientific world that their established idea was wrong. And I have to add here that such resistance is actually very appropriate. At the time, little was known about the reliability of this new technology. But then, as molecular analyses became better, and they continually reconfirmed the close phylogenetic status of fungi with animals, it became impossible to deny that this was an accurate placement. A mushroom is more closely related to you than it is to a plant. Now, in the context of the end of our previous lecture, one piece of morphological evidence that could further support this would be to show that fungi have a single rear-attaching flagellum, as this would put them firmly in the opisthecont clade. Now, this wasn't an easy thing to establish because the vast majority of fungi have no flagellated stage. How could you determine the flagellum attachment position if you don't have any members with flagella? Well, it turns out that one fungal clade, the Chytridiomycota, does have flagellated zoospores, and true to form, they push the cells from behind. This was not a characteristic that people paid much attention to before, but now that we were looking for commonalities for this cluster of seemingly disparate taxa, the animals and the fungi, it leapt out as something we could use to distinguish them from everybody else in the eukarya. The chytrids are also the most basal clade in the kingdom fungi. And this suggests that the rear flagellum characteristic is the primitive state for all fungi, with flagella being lost altogether along the other side of the basal split, the one leading to the rest of the fungi. But chytrids, they do more than just support our understanding of eukaryote evolution. They're also a likely cause or contributor to the amphibian catastrophe that's going on right now, a massive and terrifying decline of frogs and salamanders that's been taking place over the past 50 years, worldwide, but most dramatically in the Americas, the Caribbean, and Australia, and Fiji. Dozens of species have already gone extinct, and hundreds have become critically endangered over this period. And the Chytridiomycota may be the cause. Batrachochytrium, which is a genus of chytrid fungus, seems to be either the primary culprit or one agent interacting with other causal factors in creating this amphibian catastrophe. The next most basal fungal clade is the Zygomycota. And this is a fungus you've probably experienced if you've ever had a loaf of bread or some other starchy food going moldy, first becoming a large volume of cottony white filaments and eventually turning black. When this happens, it's almost certainly rhizopus, black bread mold. And if you're enough of a hippie to have ever eaten tempeh, that's made from soybeans that have been deliberately inoculated with another zygomycetes fungus named mucor. The fungus breaks down the beans, making them more digestible, while its filaments cement the partially digested beans into a solid mat. The next clade of fungus is the one that we associate with the endomycorrhizae, the soil fungi that penetrate into plants' roots and form arbuskles, and hence this group is identified as arbuscular mycorrhizae in your textbook. These arbuskles and the filaments that they attach to allow for the exchange of water and nutrients between the plants and their fungal partners. Now, these are the fungi that are so important as mutualists with plants, and this is especially true in tropical ecosystems, where soil has little organic content to trap and hold on to water and nutrients. The fungal filaments take on this role of trap and hold, and then pass along to the plants, whose roots they're connected to. The fungus, in turn, benefits from the sugars produced by the plant's photosynthesis and transported to the roots. Why is this more important in the tropics? Well, two main reasons. There's the organic poor status of tropical soils, which is the result of the warm and moist climate. Microbial decomposition of organics occurs at very, very high rates under warm, wet conditions, and what you have left over is mostly mineral, silt, and sand. This contrasts strikingly with the organic dominant quality of soils from higher latitudes. As I said earlier, with a soil being so organic poor means that the soil fungi with their extensive and pervasive mycelia are all that's left to trap and to hold water and nutrients that are usable by the plants. The other reason for a greater dependence of tropical plants on their mycorrhizae is the much higher density of plant life, sometimes with 5 or more layers of structure of perennial plants. You've got the understory, lower canopy, upper canopy, the emergent layers of really, really tall trees, as well as the lianas. These plants are often packed together into a very high density of boles, that's plants per square meter. At the same time, you really don't have the opportunity to create multiple levels in the root systems. And consequently, the root-shoot biomass ratio in these tropical forests tends to be low, much lower than what you find in temperate forests, where there's often only a single canopy layer with no understory. The plants of tropical forests with less root system are only able to maintain an adequate uptake of water and minerals with the help of arbuscular mycorrhizae. The remaining clade of crown fungi is comprised of 2 subclades of what I call fancy fungi, which includes most of the fungal organisms you're familiar with, as food, poisons, or sources of sometimes recreational chemicals. These 2 clades differ primarily in the way that their sexual spores are carried, either in asci sacs or in basidia clubs. Both Ascomycota and Basidiomycota come in all 3 growth forms: unicellular yeasts, molds, and a more complex form that spreads mycelia diffusely through a food source, and then forms some sexual phase called a sporocarp, like a mushroom, which is a fruiting body that has one main function: to disperse the sexually produced spores to potential growth sites in new locations. The basidiomycetes form basidiocarps, which are usually recognizable by the gill-like structures, usually on the underside of a mushroom cap or a shelf fungus. The ascomycetes form ascocarps, which are often cup-shaped, and hence the ascomycetes go by another name, the cup fungi. But ascocarps may also be brain-shaped, like morels, or sometimes even potato-like and smelly, as is the case with truffles. At this point, I could go on with more fungal biology, but I'm going to leave it here because there's a lot of plant biology that we have to cover in the next sequence.